Okay, looks like it is. So just for reference, today is Wednesday, March 22, and we stopped on Monday with this slide talking about the three categories of formed elements in the blood. And do you remember so we have three categories, only one is a true cell. Do you remember which category? White blood cells are the only true cell. Good. So we're going to move forward. There we go. So we're going to talk about the first category, which is platelets. And this is written on the board, nice and messy, but it is there. So platelets, very important with the clotting process. It's basically just like a sack of chemicals that are important with the clotting process. They also help with patching up damaged walls, to stop the bleeding process. They work in conjunction with the red blood cells and the fibrin fibers to help patch up the walls. And interestingly enough, this is at the very end, we may talk about this, but they also have chemicals that cause the vessels to contract. It actually starts with a vasospasm and then it contracts to help reduce the amount of blood that you lose. So there are really important functions with the platelets. So, as I mentioned, I don't know if I mentioned this part, but they are fragments of a large cell. The cell is called a megakaryocyte. Let me change the color here so I can write that down for you. So the platelets are formed from, let's see, and if I spell it wrong, sorry, karyocyte, big cells. I have a picture coming up later. So they're fragments that have broken off from this megakaryocyte, and they just are a sack of chemicals. And so they have cytoplasm from that large cell, and then the membrane, obviously, to hold it all together. And the chemicals that are within there are the ones that are necessary to start the whole clotting process. But clotting is actually kind of a sophisticated process. There are many chemicals involved, and there is a chain reaction that actually needs to occur. And a lot of clotting factors that are involved all of those clotting factors have to be functional and present in order for clotting to actually occur. But platelets is the first, it begins that whole clotting process. And so one of the important things that it does is that when a platelet bursts open and releases those chemicals, it triggers that conversion from fibrinogen, which, as a reminder, is one of those major proteins that we find in our blood that's at a fairly consistent rate all the time. It triggers the conversion from fibrinogen into fibrin, those sticky little fibers that basically form kind of like a spider web over the damaged area. And then those red blood cells stick to it, platelets stick to it, and it forms a clot, forms beginning of the scab. Okay? So remember that platelets are what triggers that conversion from fibrinogen into fibrin. I have a better picture of platelets on the next slide. So here, color changed, my bad. So here is a bunch of platelets. And so they just look like little tiny particles. And so for reference, we have a red blood cell here and here. So they're small. So if you looked at them in a microscope, they're just like little purple dots because they're just tiny little sacks of chemicals. Okay? And they only burst open and release their chemicals against a rough or a sharp surface. And so when you have an injury, even if it's like a paper cut and it looks like a clean cut, microscopically there are rough edges. And so when platelets, when you're bleeding, platelets go over those rough edges, it causes it to burst open and release those chemicals. So, so blood flows over, breaks open. And so this is the beginning. So releasing the chemicals from the platelets is the beginning of the entire clotting process. Okay, and remember, it is a chain reaction that involves many different clotting factors. And we'll talk about briefly what happens when any one of those clotting factors is not functional. Talk about that in a minute. Okay, well, it's right there, but I wanted to talk about the disorder. So if any one of there are many clotting factors, there's more than 8 of them. And if any one of those clotting factors is not present or is not functional, usually from a mutation or a genetic, sometimes it's genetic, any one of those factors not functioning, it disrupts the entire clotting process and clotting will not occur. So every single step relies on all the other clotting chemicals. Okay, so you have in your handout, in one of the handouts provided on Moodle, shows the clotting process and all of the factors involved. You do not need to know all of the factors, but you do need to know that there are many clotting factors involved with the clotting process. And as I mentioned, if any one of those is not functioning properly, it disrupts the entire process and their blood will not clot. So one disorder that deals with a person's inability for blood to clot is called hemophilia. And this is a genetic disorder. It's carried on the X chromosome. Let me make sure I'm saying that right. Yep, it's carried on the X chromosome. And so it is more common in males than females because females will only present with hemophilia if they have that mutation on both of their X chromosomes. And so it's much more common for males to have hemophilia. It's called the bleeding disease, and this is because one or more of the clotting factors is not functioning properly. One of the common ones that is affected is clotting factor number 8. There are other ones as well, and there's a mutation, and so that clotting factor, it does not function properly, so the clotting just won't work. And so what do you do when you have a person whose blood will not clot? So I will give you a personal example because my grandfather had hemophilia, and even a simple thing like bumping into a desk where we might get a little bruise, for example, a person with hemophilia bumping into that desk, instead of just getting a little bruise, it keeps bleeding and bleeding and bleeding. And you'll get this giant bruise called a hematoma. And the only way to stop that bleeding is to get a blood transfusion. Did I say that right? I think I did. Yes, sir.
Were there 8 of them that we were going to open? There's 10 in the box.
I think so. That sounds about right. Sorry, let me think back. What was I saying? I just lost my train of thought. So you need a blood transfusion from someone with healthy blood that has all those clotting factors, and so then a person's blood will be able to clot. So, example, I remember my mom telling me a story that, so my grandfather was a professor at PUC, and so if he had an emergency where he was bleeding or, he would have to be driven down to UC Berkeley Medical Center, which is about an hour and a half away from PUC, to get blood transfusions. And it still remains the major way to treat hemophilia. If you know the clotting factor that is not functioning properly, medicine has developed much more, and you can give that person an injection to stop that bleeding. So hemophilia is genetic. In fact, it's very prevalent in the royal lines in Europe. But I don't want to get into the whole history of that. But because it is genetic, it definitely passed it around all throughout Europe, which is quite interesting. So hemophilia is still happens today. We have better treatments than the past, and we also have much better ways to ensure the blood from transfusions is clean and healthy. Any of you guys donated blood before? Yeah, they go through some rigorous questioning and testing to make sure that the blood you're donating is safe, and they also test it for things like HIV. So in the past, people who were hemophiliacs, they didn't have those ways to check to make sure the blood was safe, especially for emerging diseases. So for example, my grandfather did actually get a transfusion that had HIV. And that was before they really knew what it was. And that was he probably got that transfusion sometime in the mid to late 70s. HIV really wasn't understood or really recognized at all until the mid 80s. And so there were a lot of people with hemophilia who unfortunately were infected with diseases like HIV or hepatitis, for example, from people who donated blood. It was a really unfortunate side effect, I guess, of the treatment. Okay, so more on platelets. So remember the blood vessels, they're all lined with a certain type of cell, and in the heart too. Yeah, your simple squamous epithelium, and that's nice and slick and smooth. And one of the biggest reasons for that is you want a smooth surface. You don't want the platelets to move across the rough surface and burst inside your blood vessels, because that could cause clotting within your blood vessel, and that's bad. So if a couple burst, no big deal. You're safe. But if you have many platelets that are rupturing, for example, if the inside of a vessel is rough from an infection, or, for example, the buildup of plaque or atherosclerosis in your vessels, that is more likely to cause platelets to burst and may actually form a significant clot. That is bad. Okay? So notice right here it says, so the vessel becomes rough in response to disease. You are more likely to form a clot within the body. So there are two types of clots. One is a thrombus. I'm going to write that down. Thrombus. And the other is an embolus. And again, if the spelling is wrong, I apologize. And so, by the way, again, so infections or buildup of fat, mineral salts on the vessels, again, can make it rough and more likely for platelets to burst and start that clotting process when you really don't need a clotting to occur. So we have two types of clots that will form. So a thrombus is a clot that is stuck and lodged in a vessel. Okay? That is the least dangerous of the two. So typical treatment, or first of all, typical ways that people develop these is, those long airline flights, or sitting for long periods of time without moving. Other examples like that. And you'll have pain and there's swelling. It usually occurs in the legs. And what they do is they put the person in the hospital. They keep them in the bed, restricted movement. They give them blood thinners like heparin to slowly break down that clot in a safe manner. Otherwise, if they don't do it in a nice, safe, controlled way, you can have that clot break free. So when a clot breaks free and goes throughout the cardiovascular system, we call that an embolus. Embolus, not a good thing. So when they break free, they can travel throughout the cardiovascular system, and then they get lodged wherever they cannot pass through anymore. And so this can actually be very, very serious and fatal. So example, you may have heard of a pulmonary embolism before. That is where a blood clot breaks free and gets lodged within the lungs, and that can be fatal if not treated like immediately. Another place that's very dangerous for an embolism to travel is up to the brain. So if the clot travels to the brain and gets lodged in a vessel there, the tissue past that point is not getting the oxygen it needs, and it dies very quickly. And so that is one type of stroke. Stroke can be caused by a blockage, by a clot. Very serious situation. Okay, moving on. So we're moving to the next category, to the white blood cells. Remember, the white blood cells are the only true cell in the formed elements category. So remember, formed elements, we have platelets, white blood cells, red blood cells. So I abbreviated those on the board. So white blood cells, WBC. So WBC, oh, stop that, go away. Is a very common abbreviation for that. Another term for white blood cells is leukocytes. So I'm going to write that down to leukocytes. Okay, so two different terms for white blood cells in general. So remember, they're the only true cells, and we're going to go through what these cells do briefly and look at pictures, because you will, on your exam next week, need to be able to identify the major white blood cell types, and I will give you the information you need to do that in just a moment, okay? And since this is actually recording and working, I'll be able to put it up and you guys can refer back to this. So we have a lovely list here of different types of white blood cells. So we're just going to start here. Neutrophil. Neutrophil is involved with phagocytosis, so engulfing things that are infected or old cells that need to be recycled. Also involved with the inflammatory response. Monocytes. Monocytes, even wrote it on the board. Monocytes are essentially immature macrophages. And so, again, we'll talk through this more as we go look at each picture. And so it's able to pass through the capillary walls into the tissues to it's called immunological surveillance. So they're moving throughout the tissues, looking for infected cells, looking for cells that are old or need to be recycled or pathogens, and then they engulf them by phagocytosis and break them down. Macrophage is the mature monocyte. So macrophages is what we find in the tissues. A monocyte is still within the bloodstream, so when the monocyte moves out into the tissues, it becomes a macrophage. Basophils, right here. Basophils and mast cells both are heavily involved with the inflammatory response. These are usually the ones you would blame for your seasonal allergies, because they contain histamine, which triggers that inflammatory response in response to a foreign substance, for example, like pollen or dust or mold. And so histamine is released. The inflammatory response is supposed to be a good response, bringing more blood to that area and white blood cells and helping to fight whatever foreign thing is there. But sometimes that inflammatory response goes too far, okay? And we'll talk about that more later. Then we have what's called an eosinophil, and these are also involved with allergies, but notice they inhibit inflammation. And in addition, the eosinophils are more special white blood cells and more often triggered and function in the presence of parasites. Okay? And then the last one is my favorite, natural killer cell. It's like the James Bond of the immune system, licensed to kill. Okay? And so the natural killer cell looks for any infected cells, but it it is very specialized. Tumors and virus-infected cells. So more often than you probably think, you may have an abnormal cell that occurs. But if your immune system is functioning right, the natural killer cells find that, kill it, that's it. When an abnormal cell starts to grow out of control, then you get cancer. So natural killer cells is one of the checkpoints that helps to prevent cancer from growing, or any abnormal cells from growing. So it's a very important type of white blood cell. And then it also helps kill cells that are infected by viruses. So I just love this one. Natural killer. The name is awesome. This is just an illustration of some of the white blood cells. I'm not going to use this drawing. It's just kind of a summary picture. So we're going to be looking at actual pictures of white blood cells. Forgive me, I need to sit down. All right. So first of all, I want to let you know that white blood cells are broken up into two categories. And so it's here on the board. And the two categories are granulocytes and agranulocytes. So essentially, when you're looking at the cell, it has granules in it. And it should be easily visible. Or if it does not, then it's agranulocytes. So there's two categories to keep in mind. So maybe let me back up here and I'll write that down so you have two categories. One, granulocytes, and then agranulocytes. Okay, so neutrophil is the first one we're going to be looking at. So unique characteristics of neutrophils is they have the appearance of multiple nuclei. They don't actually have multiple nuclei, but the nucleus is kind of weirdly shaped, and you're viewing it from just a single plane, and so that's why it looks like you have more than one nucleus, but it is all just one. So yeah, here it looks like you have 3 nuclei. It really is just one. For reference, you can see the red blood cells. Note that they are smaller than the neutrophil. The neutrophil is a granulocyte. So you can see the little grains in here. You can see them here as well. So if you see a picture of a white blood cell that appears to have more than one nucleus, and they're kind of this weird shape, and granules as well, and the granules are not super dark. You'll see examples where they're much bigger. This will help you. So the appearance of multiple nuclei, and remember, it doesn't have multiple nuclei, it just looks like it does, and it's a granulocyte. Okay. Next is the monocyte. And monocyte has this really large nucleus. Okay. And the nucleus kind of has a U shape to it. Looking at the picture, would you say it is a granulocyte or an agranulocyte? Do you see granules in there? Not really. So this is an agranulocyte. So those are the two main features that you're going to look for, is a white blood cell with a large nucleus for the size of the cell, and that nucleus kind of has like this curved or U shape to it. Okay? So this is definitely one that you would need to know. So maybe I need to back up and make stars for the ones you need to know. Okay. You have to identify neutrophils, monocytes. And so here's our next one, macrophage. This one I will not ask you to identify because the pictures they're just kind of ambiguous. If I did, it would be extra credit, and it would be like these scanning electron microscopy pictures. I like these two pictures because it actually shows the macrophage in the process of engulfing cells. So phagocytosis, actually in the process of engulfing them. So that's what the macrophage does, is it takes in infected cells, old cells, broken down cells inside, and then breaks them down to their components. It's a very important job. For example, red blood cells, their lifespan is only 3 months. So we have to be able to break those down and recycle those components and reuse them. Okay, next we have the basophils and mast cells, and they look very, very similar. So in fact, I'm only going to ask you to identify basophils just because they're so similar, okay? And these, are they granulocytes or agranulocytes? What do you think? Granulocytes, yeah, and they got big granules, and it's packed full. And in those granules is the histamine. Okay? Histamine is released, causes that inflammatory response, also is responsible for those seasonal allergy things like the runny nose, itchy nose, eyes, all that fun stuff. So for example, Benadryl, do you know their slogan? Do you remember their slogan? So Benadryl is the histamine blocker. So Benadryl helps prevent the histamine from causing this inflammatory response in a person's body. So just for reference, notice, okay, so this is pointing at a basophil, and here's a mast cell. It's really very difficult to tell the difference between the two, which is why I will not ask you to know the difference between the two because it's just so similar. But you should be able to know that it's a granulocyte. I will ask you just the basophil just to be consistent. Okay. Eosinophils. Eosinophils. Granulocyte or agranulocyte? Granulocyte. Yeah, look at these guys. It's packed full. So these are definitely granulocytes, and we have an interesting shape to the nucleus. So granulocyte, site, and the nucleus, it's kind of that U shape again, but it's smaller. It's not taking up most of the cell. It kind of has this narrow portion to it, and so you kind of have that U shape. It's smaller than we would see in the monocyte, and it has granules in it. So just for reference, if we look over here, there's a neutrophil. So you can see those kind of, it looks like we have 3 nuclei in that. And then for reference, if we look at the eosinophil, yeah, it looks like there's multiple nuclei, again, because the nucleus has kind of a strange shape to it. But the big difference is the presence of granules, okay? So granules, it's an eosinophil. No granules, it's going to be your neutrophil, okay? Oh, let me put a star by that because, yes, that is one that I would ask you. Okay. Natural killer cells, again, yes, this is my favorite. I have been known to ask that just for fun or extra credit. Natural killer cells, so remember tumor and virus infected cells. And this works by basically killing the cell, lyses the cell. I won't ask you to identify this because it's very difficult to do, but you should know that what it does, so tumors and virus-infected cells. So, a second, sorry. What's really neat about white blood cells is they don't stay in the circulatory system, okay? They have the ability to squeeze out through the capillary walls into the tissues, and that's really actually very important, especially if you have an infection in the tissue. You have to have a way to get those cells out of the blood to that source of infection. And so what they do is they do it through a process called diapedesis, and this only can happen in the capillary beds because that's where you have one cell thick. And if you remember, in the most capillaries, except for the central nervous system, they are fenestrated. And so fenestrated capillaries have those little gaps that allow substances to pass through as they are supposed to do. And so white blood cells squeeze themselves through, as shown in this picture. That process, remember the process is called diapedesis. That's a term I expect you to know. Okay, diapedesis right there. And so they can enter the tissue or go back into the bloodstream. So two important functions of white blood cells. So they are part of the immune system. So we will talk about their role in the immune system in a bit. Probably won't get there today, and that's okay. And so, for example, defending your body against pathogens, microorganisms like parasites, help with certain toxins, foreign substances, even its own body cells that are needing to be broken down, whether they're just not functional or old. And then also secondly, kind of as a custodial crew or a cleanup crew. So as an example, red blood cells, their lifespan is about 3 months. So after, 3 months, those red blood cells start getting old, they're not functioning. White blood cells, especially macrophages, for example, take those in, break them down to their components that can be recycled and make new blood cells. So recycling the iron, for example. So helping to keep the body clean, free of debris, as it says. And so each white blood cell does have a specific task, as I kind of summarized at the beginning. And you will need to be familiar with those specific tasks that each of those white blood cells do. So I just gave you like the Cliff Notes version of what they do. So you're going to have to read your textbook and go through that information, okay? More on white blood cell disorders. And so I know some of these questions are on your worksheet because I was getting people asking me questions about it this morning. So you must know these white blood cell disorders. I will not show you a picture and expect you to diagnose it, but I do expect you to know the disorder and what is it characterized by. So for example, leukopenia. This is where the white blood cell count is really low. Okay? So way lower than normal. And what kind of effect would that have on a person? Yeah, it's going to affect your immune system. It's basically, you're going to have a harder time fighting infection. That's a bad thing. And actually, HIV, that virus, attacks white blood cells. So that can cause leukopenia. Especially in later stages. That's why people with HIV, when they start getting these infections that people would normally not get, who have a functional immune system, that transitions them into what we would call AIDS, which is an autoimmune deficiency syndrome. And so those opportunistic infections then start taking over and can kill a person, whereas a normal healthy person with enough white blood cells would be just fine. Okay, the next one is leukocytosis, and this is where the white blood cell count is just like way high. Okay, so I need to watch my words on this one. Abnormally high, okay? Because leukemia is where it's astronomically high, okay? So you do need to know these three. We'll be very careful with the wording. Leukocytosis is the white blood cell count is too high. Leukemia is like way too high, off the charts, extremely high, okay? All blood cells are created within the bone marrow, okay? And so that's why people with leukemia, very often, they'll have radiation, kill the cells in the bone marrow, and then they'll get a bone marrow transplant of healthy blood cells, and that usually will cure that problem. Okay, so more on white blood cells. You don't need to know a ton of this, but note here, all blood cells originate from one type of cell, and that is called a hemocytoblast. So I did yellow, so that's more obvious. So this is basically, I don't know how you'd say it. Yeah, so they all originate from that. Or like you could say it's the original stem cell, okay? And then from the hemocytoblast, it divides into two categories of stem cells. So you have hemocytoblast, and then it branches off to either myeloid stem cells, I can't spell, or lymphoid, okay? So I know, it's awful. So hemocytoblast is the ultimate stem cell, then branches into two categories. So myeloid stem cells is category number 1, lymphoid is category number 2. So myeloid stem cells produces all white blood cells except for lymphocytes. So myeloid stem cells do almost all of the blood cells, okay? Lymphoid stem cells produces lymphocytes. Okay, and so that production of creating or producing these lymphocytes, we call it lymphopoiesis. So that is the term for the production of making those lymphocytes. So if I asked you a question of which stem cell are most blood cells created from, you would know it's the myeloid stem cells. And if I specifically ask which stem cell do lymphocytes originate from, be the lymphoid stem cells. Okay, so most come from the myeloid. White blood cells develop in different places, so they are formed within the bone marrow, as I told you before, as are the red blood cells. Most develop and mature in the bone marrow, except for a few cases. Okay? So, except for monocytes, the white blood cells develop in the bone marrow fully. But monocytes are immature macrophages. So, these monocytes finish their maturation process in the peripheral tissue. So when a monocyte squeezes out of the capillaries into the tissues by diapedesis, it is then considered a macrophage, and that is the mature form, so that it matures in the peripheral tissue. So please remember, monocytes mature into macrophages, and that occurs in the actual tissues of your body, not the bone marrow. Okay, this illustration is also in your book, and it kind of just breaks down all the different types of blood cells and where they originate from. So if we look at the very top, the mother of all blood cells is the hemocytoblast. Then it divides into two categories, myeloid stem cells and lymphoid stem cells. Yeah? Okay, so then you can just kind of follow this. I am not going to ask you questions about this because this is very detailed, but I want you to take a look at it, just kind of appreciate how these cells develop. I did want to point out, so megakaryocyte right here, this is that big cell where fragments break off. Those fragments become your platelets. So they originate from this big cell in the bone marrow, and then we have these little fragments that break off, and those are has cytoplasm and the plasma membrane, and you have your platelets. I think that's all I wanted to mention there. Okay, finally, moving to red blood cells. What's the other term for red blood cells? Erythrocytes. Let's see if I can write that. Erythrocytes. And then we also abbreviate it as RBC, red blood cells, RBC. So know the different terms. You need to know what erythrocyte means, okay? You have to know what a leukocyte means, which is a white blood cell. These terms are important, so I can't, if you ask me on your test on Wednesday, what does erythrocyte mean, I can't tell you, okay? So as we talked about before, red blood cells are not true cells. And so the reason they are not true cells is there is no nucleus, okay? So no nucleus, no ribosomes, or mitochondria. So it is not able to do cellular respiration and make energy. It doesn't have the nucleus, so it doesn't have cellular metabolism, so it's not a true cell. So this kind of begs the question of, so when someone does a blood draw, how are they getting your DNA? They're getting it from your white blood cells, because they're not getting it from your red blood cells. They lose their nucleus in the process of maturation. Okay? And these are produced in the red bone marrow and live about 3 months or 120 days. And then, do you remember what cell recycles them? It's your macrophage. So the macrophage is what helps break down and recycle old red blood cells. Okay? Remember, they're part of the cleanup crew. That's one of their functions. Okay, so we're going to talk about red blood cell now, its shape, its function, and it has just one job. And its job is to grab onto oxygen in the lungs and carry it to tissues where the oxygen levels are low and deliver the oxygen. That's their job, okay? So, interesting thing is, so we know that in the bloodstream, we have all of these components dissolved in the plasma, like gases and nutrients and waste products, but we can't dissolve or absorb enough oxygen to supply the oxygen to all the tissues in our body just in the plasma. So we have to use these red blood cells to transport the oxygen throughout the tissues. And so that is the one and only job of red blood cells, is to carry oxygen, because we can't dissolve enough in our blood, okay? And so we use a protein called hemoglobin. Better know your protein name. Hemoglobin is a protein. It's made up of 4 protein subunits that also have an iron atom, okay? And that iron atom of the hemoglobin grabs that oxygen and holds it as it's transporting throughout the circulatory system and then releases the oxygen in areas where oxygen levels are low. Okay? So we'll go to the next one. And so oxygen, more on oxygen. We'll just make this the last slide, I believe. And so remembering the whole cellular respiration process, what were the products, or what were the things that went into the reaction? So the reactants. So I'll help you out. So we need glucose, remember this? And we needed oxygen. And then what were our products? We had water, plus what? Carbon dioxide, plus energy, ATP. Okay? So that's cellular respiration in a nutshell again. So we need to do this. We need oxygen in order to make energy. Okay? And I know you know this. We've talked about it both semesters. We need the oxygen on a constant basis to make energy so that our cells and our body can function properly. So the red blood cells carry the oxygen that we need for metabolism. Okay, so last note here, and I'm just going to read it. Look, it's bold and italicized, big hint there. The more red blood cells in the bloodstream, the more oxygen that can be delivered. So have you heard of blood doping before? Athletes do this. So they'll draw blood, they'll store it, and then before an event, they'll transfuse back their own blood into their system so they have extra red blood cells, more than normal, so they can carry more oxygen, which gives them a slight advantage over other competitors. Or the other thing they can do is train at high altitudes because you have to produce more red blood cells to get the oxygen to your tissues. Yes, ma'am?
You were saying on Monday how you had a friend that produced too many red blood cells and then she had to get rid of them?
Yes, I read about that, and I forgot the name of the disorder.
So if it's a good thing for like athletes and people, why did he have to let his blood out?
Shoot, I wish I had that file open. So I'm having a hard time remembering. You produce too many, and I'm going to have to get that article for you because I read about it. I forgot the name of it, but it's too, yeah. I'll get back to you because I did read about it, but I'm drawing blank right now. I'm sorry. Okay, guys, have a good day, and I'll see you in lab or